Good morning, everyone. My topic for today's presentation is immune system interface with psychiatry, and my moderator is Dr. Veena ma'am. This is an overview of all the topics that we will be going through today. We will be first talking about the immune system, then the social adversity, environmental challenges, and the immune response, immunological mechanisms, immune system effects on CNS, immune system and psychiatric diseases, COVID sequelae, psychiatric symptoms and immunological mechanisms, therapeutic implications, behavioral interventions, and immunity. First, we will talk about the immune system. The immune system can be divided into innate immunity and acquired immunity. The innate immunity, it includes in physicochemical barriers, it includes skin and mucous membranes. In cells, it includes phagocytes, that is macrophages, neutrophils, and natural killer cells. Then it also includes soluble mediators that affect other cells, like macrophage-derived cytokines, that is IL-1, IL-6, TNF-alpha, and IFN-alpha. In acquired immunity, the physicochemical barriers include cutaneous and mucosal immune systems. In cells, it includes B and T cell lymphocytes. And in soluble mediators, it includes lymphocyte-derived cytokines, that is IL-2, IL-4, IL-5, IL-6, IL-10, and IFN gamma. Innate immunity has no memory, while memory is present in acquired immunity. And innate immunity, it includes complements and acute phase reactants in circulating molecules, while in acquired immunity, there are antibodies. First, we'll talk about the innate immunity. The cells mediating innate immunity do not require prior activation and or specific recognition of invading pathogens to be functional. They provide an important first line of defense against infectious agents during the early stages of an immune response. This is a diagram which is showing the innate immunity. There may be local effects or systemic effects. Now, when NFKB is stimulated, it releases TNF, IL-1, IL-6, and IFN-alpha. Now, in local effects, it stimulates stromal cells to release chemokines and endothelial cells for expression of adhesion molecules and immune cell margination and diapedesis. Along with this, there is also increased vascular permeability, vasodilatation, and inhibition of viral replication. Systemic effects of these TNF, IL-1, IL-6, and IFN-alpha, they will act on the brain and they can also act on the liver. The effects on brain include fever, fatigue, anorexia, anhedonia, altered sleep, cognitive dysfunction, and HPA axis activation, while effects on liver include acute phase response with release of C-reactive protein, serum amyloid A, haptoglobin, and alpha-1 anti-chymotrypsin. In acquired immunity, the principal effector mechanisms of acquired immunity are mediated by antibodies, which is your humoral immunity, which is secreted from B cells, and by T lymphocytes that kill target cells directly, and macrophage activating cytokines such as IFN gamma, which is your cellular immunity. Humoral immunity is especially effective in combating extracellular pathogens such as bacteria and parasites, while cellular immunity is effective in protecting against viral and intracellular bacterial infection, and as with natural killer cells, may provide some protection against tumor cells. Now we will talk about social adversity, environmental challenges, and the immune response. First, we'll talk about the stress and illness. Now, stress as well as depression, through their effects on inflammation, has been linked to increased morbidity and mortality in infectious diseases, such as HIV infection, autoimmune disorders, neoplastic diseases, as well as diabetes and cardiac illness. Acute stress exposure Acute stress exposure in humans and lab animals has been shown to increase the expression of innate immune cytokines, activate microglia, and sensitize subsequent immune responses to inflammatory immune challenge. Now, chronic or severe stress, like acute stress, chronic stress also appears to be associated with enhanced pro-inflammatory activity. Enumerative changes such as increase in circulating white blood cells and decrease in natural killer and T cells, as well as alterations in number and ratio of CD4 plus and CD8 plus T cells have also been reported. Recent data indicates that early life stress, including parental loss and or physical or sexual abuse during childhood, is associated with increased markers of innate immune system activation, including CRP, in both non-depressed and depressed individuals at rest and in response to both acute and chronic stress. Now, factors which increase inflammation and increase risk of depression are early life adversity, obesity, sedentary lifestyle, processed foods, psychosocial stress, social isolation, poor sleep or insomnia, smoking, drug abuse or dependence, low socioeconomic status, female sex, and reduced childhood exposure to co-evolved microorganisms, while factors which decrease inflammation and decrease risk of depression are Mediterranean diet, exercise, positive social connectivity, early contact with pets and farm animals, experiencing reverence and awe, antidepressants, electroconvulsive therapy, psychotherapy, and meditation. Now, this diagram shows stress and bidirectional neural or immune interactions. Infection, tissue damage, or destruction can cause stimulation of NF-κB, which causes release of TNF, IL-1, and IL-6, which causes inflammation, which includes increased pro-inflammatory cytokines, increased chemokines, increased adhesion molecules, and increased acute phase reactants. Now, these molecules, they enter the brain via leaky passages in the blood-brain barrier or via sensory vagus or via active transport molecules, and they are involved in pathways which are responsible for depression, such as altered metabolism of serotonin and dopamine, altered metabolism of of brain-derived neurotropic factors, and they also stimulate corticotropin-releasing hormone in paraventricular nucleus and cause release of glucocorticoids. Now, all of these, they are involved in the pathology of depression. And now stress, it stimulates NF-κB by release of pro-inflammatory sympathetic nervous system responses, such as release of norepinephrine, and while also it can also inhibit your vagal input, that is your acetylcholine release, and hence it can increase inflammation. Immunological mechanisms. In general, a stressor can alter immune function in two major ways. First, the stressor can lead to changes in the distribution of immune cells in any given part of the body. Second, stress can alter the function of the immune cells themselves. Now, in neuroendocrine mechanisms, it includes the sympathetic nervous system and the HPA axis. Now, in sympathetic nervous system, catecholamines have been shown to stimulate the production of pro-inflammatory cytokines, especially IL-6, in both the peripheral blood and CNS, and activate inflammatory signaling cascades in peripheral blood mononuclear cells. Activation of alpha-1 adrenergic receptor may be involved in innate immune system activation following norepinephrine exposure. Regarding the effects of stress on acquired immune response, catecholamines via activation of beta-2 alpha receptors, beta-2 adrenergic receptors have been shown to inhibit Th1 cytokines and promote T cell and inflammatory responses. Now, HPA axis, it serves as a central component of the mammalian stress response system and ultimately functions to maintain bodily homeostasis via mediation of immunosuppression and immune system activation. This is exemplified by varied effects of both corticotropin-releasing hormone and glucocorticoids. Corticotropin-releasing hormone applied within the CNS, it suppresses several measures of immunity, including mitogen-stimulated lymphocyte proliferation, antibody formation, as well as T cell responses to T cell receptor antibody. In contrast to its immunosuppressive properties, corticotropin-releasing hormone has also been shown to enhance pro-inflammatory cytokine production in humans when administered peripherally or within the CNS. Glucocorticoids modulate the following immune cell trafficking throughout the body, cell death pathways, Th1, Th2 cellular immune response patterns in a manner that inhibits Th1, that is the cell-mediated responses, and promote Th2, which are your antibody responses. Now, immune system effects on the CNS. First is cytokine effects on behavior. When challenged with a medical illness or chronic psychological stressor, complex interactions between the immune and nervous system promote immune-induced behavioral changes referred to as sickness syndrome or sickness behavior. It includes dysphoria, anhedonia, fatigue, social withdrawal, hyperalgesia, anorexia, altered sleep-wake patterns, and cognitive dysfunction. Cytokine effects on the brain. Pro-inflammatory cytokines released in the periphery are capable of rapidly affecting CNS function. Studies have now shown that release of TNF from inflamed peripheral tissues stimulate microglial cell production of chemokine ligand 2 that then attracts monocytes to the brain. Cytokines mediate their effects in the CNS through their receptors that are expressed throughout the brain and with especially high concentrations in brain regions involved in the regulation of affect, stress responsiveness, and learning and memory, including the hippocampus, amygdala, striatum, prefrontal cortex, and hypothalamus. Now, cytokines, neurotropic factors, neurogenesis, and neurodegeneration. Physical and psychological stressors suppress neurogenesis in the hippocampus, promote apoptotic cell death, and reduce the density of synaptic connectivity between nerve cells. Pro-inflammatory cytokines, including TNF-alpha, IL-1, and IL-6 within the CNS, have also been shown to play key roles in neurodegenerative changes found in illnesses such as Alzheimer's disease, multiple sclerosis, and HIV dementia, as well as following a variety of insults such as ischemia, trauma, and radiation exposure. Cytokine effects on neurotransmitters relevant to serotonin metabolism. Activation of enzyme indoleamine 2,3-dioxygenase, which is activated by IFN-alpha, is believed to be involved in cytokine-induced neurotransmitter alterations, in part by diverting the metabolism of tryptophan into kynurenine. Kynurenine can be further converted into the neurotoxic metabolite quinolinic acid by activated microglia and infiltrating monocytes and macrophages in the brain. Increasing evidence implicates kynurenine and its metabolites in depression. IFN-alpha has been shown to lead to altered metabolic activity in brain regions high in dopaminergic neurocircuits, including the basal ganglia, and relevant to cytokine-induced activation of indoleamine dioxygenase. Data indicate that intrastriatal administration of kynurenic acid, a breakdown product of kynurenine, dramatically reduces extracellular dopamine in the rat striatum. Cytokine effects on neuroendocrine system. Inflammatory cytokine effects on HP axis include increased production of corticotropin-releasing hormone and cortisol, and decreased tissue sensitivity to glucocorticoid hormones. This increase in corticotropin-releasing hormone activity may contribute to cytokine-induced depression or sickness behavior. Now, immune system and psychiatric diseases. First, we'll talk about major depression. For many years, major depression was seen as a quintessential example of how stress-related disorders may decrease immunocompetence. Studies now indicate that, in addition to immunosuppression, major depression is also frequently associated with inflammatory activation. Effects on acquired immune or lymphocyte responses Enumerative immune changes shared by major depression and chronic or severe stress include a decrease in lymphocytes, B cells and T cells, and decrease in ratio of CD4 plus to CD8 plus T cell subsets. Available evidence suggests that depression may impair T cell function in ways relevant to disease vulnerability. Because major depression is a heterogeneous condition, immune changes are not uniform across all patients, and in general, immunosuppression tends to be most robust in patients who are older, hospitalized, or have more severe and or melancholic types of depression. Effects on innate immune inflammatory responses. There are reliable associations between depression and increased plasma concentration of IL-1B, IL-6, TNF, and CRP. Increased inflammation, which is measured as either CRP or to a lesser degree IL-6, predicts the future development of depression, thereby increasing the probability that inflammation does represent one pathway for depression, even in individuals who are otherwise medically healthy. Given the role of IL-6 and CRP in predicting disease outcome in both cardiovascular disorders and diabetes, the relationship between depression and activation of innate immune inflammatory response may explain the negative impact of depression on a number of illnesses. It may also explain the fact that depression predicts later development of a range of medical illnesses, including cardiovascular disease, stroke, diabetes, and dementia. Now, bipolar disorder. Several studies have observed that bipolar patients, especially when manic, demonstrate increased plasma concentrations of soluble receptor for Th1 promoting inflammatory cytokine IL-2. Bipolar manic patients have also been reported to have increased concentrations of IL-8 and TNF-alpha, as well as acute phase reactant CRP. Several studies indicate that treatments for mania, such as lithium, lower plasma concentration of a number of cytokines. It should not be surprising that mania should be associated with increased inflammation, as mania and depression have also been reported to show identical neuroendocrine and autonomic abnormalities, such as dexamethasone, non-suppression, and increased sympathetic activity, both of which would be expected to promote inflammatory activity. Now, schizophrenia, while inflammatory activation in adults does not routinely produce psychosis, substantial animal data as well as powerful circumstantial evidence in humans suggests that increased inflammation very early in life, especially in prenatal and perinatal periods, promote changes in CNS development and function, has significantly increased the risk of manifesting schizophrenia in adulthood. Moreover, increasing evidence suggests that reducing inflammation in youth at high risk for schizophrenia may prevent the transition to full-blown psychosis. Multiple studies have found associations between in vitro exposure to a range of infectious organisms like influenza virus and the development of psychosis in general and schizophrenia, particularly in adult life. Many other prenatal factors linked to an increased risk for the development of schizophrenia have also been associated with increased levels of inflammatory biomarkers during pregnancy, including obstetric complications, gestational diabetes, severe physical or psychosocial stressors, and maternal depression. Both animal and human data suggest that long-term impact on inflammatory activation on the developing CNS does not stop at birth but persists across early life. For example, large population-based epidemiological studies have found that individuals with a history of an autoimmune condition early in life have a 29 to 45% increased risk of subsequently developing schizophrenia. Although inflammatory activation only rarely produces psychotic symptoms in humans, patients with schizophrenia have been shown in numerous studies to demonstrate increasing number of immune biomarkers. Importantly, these alterations have been found in first episode patients, strongly suggesting that immune activation is associated with the disease itself rather than merely with its treatment or with other factors that ensue as the disease progresses. Now, autism. Autism has been associated with a number of immune abnormalities, including unbalanced Th1 or Th2 cytokine production, reduced NK and T cell activation, and increased TNF-alpha and IL-12, especially in patients with comorbid gastrointestinal symptoms. In addition, patients with autism exhibit increased autoimmune-based genes, including HLA, DRB104, and complement C4b null. The claim that autism is triggered by childhood vaccines has not been substantiated by recent epidemiological studies, and immune-based therapies for autism have not been reliably effective in support of a positive role for the immune system in at least some cases of autism. A study from Denmark found that in utero exposure to maternal influenza infection was associated with a twofold increased risk in prolonged episodes of maternal fever, caused a threefold increased risk of infantile autism. Now, in other disorders, increasing evidence suggests that increased inflammation is a hallmark of nearly all types of psychiatric disturbances. No currently defined psychiatric condition has been reliably associated with reduced levels of inflammatory biomarkers. Many studies now report that PTSD is associated with increased peripheral inflammatory biomarkers, regardless of whether PTSD results from civilian or military trauma. There also appears to be a spectrum of pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections, presumably as a result of anti-neuronal antibodies that develop following infection with group A beta hemolytic streptococcal infections, as exemplified by Sydenham chorea. Children with Sydenham chorea frequently exhibit obsessive compulsive symptoms, emotional debility, and hyperactivity. Moreover, children with repeated streptococcal infections exhibit higher rates of distal coriform movements and behaviors consistent with ADHD. And recent data also suggests that OCD more generally may also be associated with increased levels of a number of cytokines, including IL-2, IL-4, and TNF. Now, COVID sequelae, psychiatric symptoms, and immunological mechanisms. Survivors of COVID were clinically diagnosed with PTSD in 54.5%, depression in 39%, pain disorder in 36.4%, panic disorder in 32.5%, and obsessive compulsive disorder in 15.6% at 31 to 50 months post-infection. A subset of COVID-19 cases are associated with a hyperinflammatory response, and immunomodulatory therapies have therefore been proposed in the treatment of severe cases. High doses of corticosteroids were administered to treat SARS-CoV-1 infection symptoms during the acute phase, but were associated with organic hallucinations and manic symptoms. There is also ample evidence of adverse neuropsychiatric symptoms following corticosteroid therapy, impacting about 35% of treated patients, including cognitive and sleep disturbances, delirium, hypomania, mania, psychosis. And these neuropsychiatric effects are typically acute and resolve upon treatment. Fluoxetine, fluvoxamine, they are SSRIs, which act as lisinotropic agents and inhibitors of acid sphingomyelinase and lysosomes, and as ligands of sigma-1 receptors, mechanisms that might be used to fight severe outcomes of COVID-19. They may also exert the protective effect against severe COVID-19 through the downregulation of inflammatory response induced by viral infection. They interfere with platelet activation and aggregation. Therefore, they can potentially increase bleeding time and reduce neutrophil recruitment and inflammation, which is therefore of some benefit in advanced COVID-19 and counteracts the hypercoagulable state of platelets observed. Now, in therapeutic implications in antidepressants and immune system, several studies in humans suggest that antidepressants are able to ameliorate mood disturbance in the context of chronic cytokine therapies, especially if given prophylactically prior to cytokine exposure. For example, SSRI paroxetine significantly decreased the development of major depression in patients receiving high doses of IFN alpha for malignant melanoma. Many studies also indicate that in general, antidepressants decrease immune system responsivity in ways that are likely to be of benefit in the context of immune activation. Concomitant with attenuating pro-inflammatory cytokine production, antidepressants enhance production of anti-inflammatory cytokine IL-10. Now, antidepressants also enhance activity in intracellular second messenger systems, which are known to suppress production of pro-inflammatory cytokines and also reverse the relative glucocorticoid resistance, which is found in many patients with major depression, and suggesting that antidepressants may diminish activity in CRH and immune pathways by restoring normal glucocorticoid-mediated feedback inhibition. Now, immune modulators in psychiatric illnesses. Now, a logical correlate to the idea that antidepressants modulate inflammation is that agents with the capacity to directly diminish cytokine production may be of value in stress-related conditions such as depression. And in animal models, direct injection of SIL1 RA, which are endogenous inhibitors of pro-inflammatory cytokines, blunts HPA axis responses to psychological stressors such as distress. Studies of NSAIDs as augmenting agents for SSRIs in major depression are either methodologically weak or have failed to be replicated, and epidemiological data suggests that while some NSAIDs may be of benefit, others may actually increase the prevalence of psychiatric symptoms. Now, in behavioral interventions, they may have a beneficial effect not only in terms of mitigating the effect of stress on immune functioning, but perhaps also in diminishing emotional disturbances that arise in context of immune system dysregulation. Two factors that have been repeatedly identified as protective against stress-induced immune alterations are social support and ability to see stressors as being to some degree under individual's control. Now, the two types of psychotherapy most commonly examined in illnesses associated with immune dysregulation are group therapy, which provides social support, and cognitive behavioral therapy, which provides cognitive reframing techniques aimed at enhancing one's sense of agency and hence control. In addition to treating depression, these therapies have also been shown to diminish IF and gamma production in patients with multiple sclerosis over 12-week treatment course. Such interventions may limit the impact of stress on immune response and may have direct effects on neuroendocrine immune functions. These are my references. Thank you.